Okay. First of all, welcome everyone, and thanks for coming to this, the sophomore edition of the Bernice Grafstein Lecture in Neuroscience within the integrated program in neuroscience. And to initiate the proceedings, I'd like to invite Sienna Drake, who is our president of our very active and commendable student association to the podium.
Thanks, Joe. So I just want to welcome you all to the second annual Bernice Grafstein Lecture. As Joe said, my name is Sienna, and I am the president of the Graduate Students Association for Neuroscience this year. So before we get started with the main talk by Dr. Tessier-Lavigne, we would be remiss to not acquaint ourselves with the person who has inspired this annual lecture series. So Dr. Bernice Grafstein is a remarkable scientist, and I get the honor to tell you exactly why that is for approximately the next 3 to 5 minutes. So her illustrious career began with an honors degree in biochemistry and physiology at the University of Toronto, and she pursued her PhD here at McGill, where she characterized the mechanism behind a phenomenon known as spreading depression, which is known to be involved in seizure and migraine. And her work on this topic was so well regarded by her PI and appreciated that her supervisor actually let her publish it under her name alone. And these articles also remain highly cited even today. So next, she pursued a postdoctoral position at the University College London in the Department of Anatomy, and here she characterized the connections between the corpus callosum and the cortex. And following her postdoctoral research, she was invited back to McGill for a professorship, and then she moved to Rockefeller University, and after many successful years of research there, she ultimately landed on her feet at Cornell University, where she still remains teaching medical students today. So her work in these years on axonal transport and axon regeneration has helped shape the field as we know it. This included the discovery and characterization of fast axonal transport, and as well she demonstrated the transfer of cellular material between axons of the retina and also of the cortex, which, as many of us know, was critical for the discovery of ocular dominance columns. And her involvement in a variety of academic committees and conferences on the topic of spinal cord injury and axon regeneration led her to become the first female president of the Society for Neuroscience. So now that we're all on the same page about Dr. Grafstein's career highlights, I would like to get into the details of what actually makes her remarkable in my eyes, the things that you aren't likely to find listed on her Wikipedia page. So throughout her career, Dr. Grafstein has embodied 3 essential qualities of any research scientist: determination, curiosity, and balance. Her determination to study the nervous system began in her undergrad, and this led her to find a very obscure neurophysiologist to do a summer research position with, despite the lack of neuroscience topics covered in her undergraduate degree. And then within this time, she was determined to pursue a PhD in neuroscience as well, which brought her here to McGill. And this propelled both her PhD and postdoctoral research, gaining her much recognition among her scientific peers. She was invited to participate in conferences and congresses, and even during her PhD, she was asked to give a live demonstration of her cortical preparations that she used for her electrophysiological recordings, which she used to characterize the mechanism of spreading depression. And this was at the International Physiology Congress held here in Montreal. Her innate curiosity about the brain propelled her career through various topics in neuroscience, from the connections of the brain itself to axon targeting and optic nerve regeneration in the goldfish. And her curiosity and determination actually moved her to apply for an educational course in developmental biology during her first professorship so that she could switch from studying the connections of the brain and go into something more developmental and regeneration-wise. And she was able to afford this due to the generosity of the Grass Foundation, of which she is now a lifetime member of the Board of Trustees. This opportunity allowed her to ask new questions about nerve development and regeneration that she couldn't previously answer with her history in electrophysiology. And lastly, and perhaps most importantly to note for us graduate students as we spend all our time here in lab, is that Bernice Grafstein maintained balance throughout her career. During her undergraduate degree, she was the program director of the University of Toronto Film Society, and during her graduate degree, she tried her hand at radio, where she did book reviews of science fiction novels. So her involvement in various arts communities led her to become friends with painters, film directors, a ballerina, and even, she says in her autobiography, an Arctic explorer. So I'm curious to know what that exactly entails. And when she obtained her professorship at Cornell, she negotiated into her contract that she wasn't required to work Saturdays, which had previously been the rule up until that point for professors at Cornell. And when she had her first son while he was young, she didn't take lectures that required her to be away from home overnight so that she could spend more time with her family. Overall, this highlights her awareness of the importance of leading a balanced life, both by maintaining time with friends, with your artistic pursuits, and with her family. So there's no perfect way to conclude this introduction, but only to hope that I've expressed to you what I've found to be most remarkable about Dr. Grafstein in the time that I've gotten to know her. I leave you with this, a final sentiment quoted from her autobiography. I often set out to do things that I was little prepared for, but determined to pursue my own path, the path that felt right for me and not necessarily the path that the world around me would have accepted as appropriate. So I'd like to thank you again, Dr. Grafstein, for your philanthropy in this event and through the Grass Foundation, which has helped fund the Neurosymposium held by the graduate students across Montreal in the past. And in the spirit of this event, to encourage intellectual collaboration among its attendees, I would like to thank you for your commitment to in your own life, to curiosity, and to encouraging it in others, bringing us all here together today. So thank you.
Thank you, Sienna. I'd now like to invite Fred Charon to the podium to complete the MC portion of the event and, in fact, to introduce our keynote speaker. Fred?
Thanks, Joe. Good afternoon, everyone. So I'm Fred Charon. I'm a professor at Université de Montréal. I'm also a lab director at Montreal Clinical Research Institute, IRCM, and also adjunct professor at McGill. So it's my real pleasure today to be introducing Dr. Marc Tessier-Lavigne. Dr. Marc Tessier-Lavigne, as many of you know, was my mentor, my advisor during my postdoc. And that's something I'll get a bit more on later. So before that, let me just briefly summarize Marc's career. So Marc is at first a product of McGill. So Marc was an undergrad student here at McGill University. He did his undergrad studies in physics. After that, Marc did a PhD and a postdoc. He was actually a Rhodes Scholar, and started his lab in 1991 at UCSF, University of California in San Francisco. Shortly after that, Marc became an Howard Hughes Medical Institute investigator, and in 2001 was recruited to move to Stanford University, where he was a full professor. Shortly after his move to Stanford University, Marc was recruited to Genentech, one of the biggest biotech in the world, to become senior vice president, and eventually became CSO of Genentech. In 2011, he became president of Rockefeller University in New York, and in 2016, so a couple years ago, he became president of Stanford University, which is his current job. One thing that I want to emphasize is that all of this was done while still maintaining a very productive lab, and he still maintains nowadays also a lab. So this is remarkable, right? What looks like already super big job for someone, Marc was able to do this in parallel to still running a very productive lab. He also had many other jobs that he was doing in parallel to this on the board of many companies. He was also on the editorial and still is on the editorial board of many journals. So Marc is basically someone which we can say is very busy. But Marc's ability to really cope with all these things and multitask between all these things is really remarkable. So, of course, Marc has had so far an extremely productive career. He's published over 250 papers. Importantly, these papers a lot of them are published in top journals, but even more importantly than that, the discoveries that Marc has brought, especially to the field of axon guidance and axon regeneration, is enormous. A lot of Marc's work is now what we find in book chapters in textbooks that are used to teach undergrads, for example, on the development of the nervous system. So Marc has brought many concepts to our understanding of developmental neurobiology nowadays. So maybe if I have to name one of the most famous discovery of Marc, I guess, and one which is dear to the MNI here also, is the discovery that Nephron are chemoattractants for developing axons. And this is work that was done in Marc's lab early on when Marc was starting his lab. And this was a huge biochemical effort that led to the identification of the Nephron proteins. Of note, Tim Kennedy at that time, who's here a PI at the MNI, Tim Kennedy was a part of this effort and spearheaded actually one of these two Nephron papers. So this was a heroic effort, which Marc will even mention a bit further about today. Although this was a little while back, there's still a lot to discover. There's still a lot left to discover about how Nephron proteins are functioning nowadays, and this is what Marc will be telling us more about. So Marc, in addition, also received numerous awards. And here, I'm not going to go into details of those. He's received so many awards that it would just, it would take too much time to list all of these awards. But, you can think of many of the most prestigious ones you can think about. He got it. He's also received an honorary degree from McGill University. So one thing that I want to focus on for the rest of my introduction is, as opposed to all the awards and all the other things that Marc has done, is really a more personal aspect of Marc. And this is really his ability to mentor. So Marc was a fantastic mentor for me, and he was also a fantastic mentor for a lot of people. And I think given the audience here today, given that this event is organized by IPN students, I thought that given that the number of trainees, students, and postdocs, I thought this was an important aspect of Marc that I would like to emphasize. So I think this picture here reflects really well Marc's ability to mentor. Just as when you think that some people might be unredeemable, and you might think that there's nothing to do with that kind of people, Marc's extreme mentorship can actually end up doing something good. And so as you can see here, even in my worst days, Marc's mentorship was able to bring me back to something nowadays. And in case you guys are wondering, some of you know this person here, in case who you're wondering who that is, that's also one of our esteemed colleagues at RCM. So this is just to tell you how powerful Marc's mentorship can be. He can convert desperate lost cases into things that are okay. But jokes aside, and I think I will maybe move this away so that we can you can listen to what I'm actually going to say next. So I think one really key aspect of Marc is the fact that he has trained a lot of people who are working in neuroscience nowadays and also in other fields. So Marc in total so far had 69 trainees. 50 of them were postdocs, 19 were grad students. And of these, impressively, 36, so a bit more than 50%, currently have faculty positions. And we're talking about faculty positions at McGill, at Harvard, in the UC system, at Duke, you name it. Basically, some of the most prestigious institutions in the world. So this is really remarkable. And don't get me wrong, the other ones that didn't become PI were also extremely successful people. And so, for example, some of you might know Katja Brose, who was the editor-in-chief of Neuron for many years, who's now working with the Chan Zuckerberg Initiative, and other people like Andy Plum, for example, and other people who've become leaders now in various biotech and pharmaceutical companies and other places. And one of the things that is important to mention is that Marc was an amazing mentor, but for two reasons. One reason is obviously the mentorship that Marc can give one-on-one, which I would call a vertical type of mentorship, where your mentor is directly giving you advice. But there was also in Marc's lab a much more horizontal type of mentorship, and some of you might have heard this expression that I've used before, where Marc was able to assemble a lab with fantastic people. And really, a lot of the mentorship that I obtained was from Marc, but also from the peers that I had. So it was a peer-level type of mentorship. that I obtained in Marc's lab. And when I talk with other people who went through Marc's lab, they will all say the same thing. The team of people that we were overlapping with in the lab was just fantastic. And we would learn so much from one another from there. And this is, I think, what constitutes a big part of the success of Marc, is his ability to assemble remarkable teams that can all work together and really progress nicely together. So on this, I would like to really thank Marc for welcoming our invitation. And I would like you to join me in welcoming Marc for his talk today.
Well, thank you very much, Fred, for that very kind and also colorful introduction. When all is done, we'll go and put our makeup on and dress up again. You're all welcome to join us at Fred's lab later today. It's really a great pleasure to be here back in Montreal, back at McGill, to see so many old friends, to meet, to make new ones as well, and to have the opportunity to tell you about our work. It's also a great pleasure to be able to give a talk that's named for Bernice. You heard earlier from Sienna about what a remarkable person and a remarkable scientific leader she is, with so many foundational discoveries, starting with her seminal discoveries on the mechanisms of spreading depression, but then pursued over many decades subsequently. I feel that all my life I've just been chasing after Bernice. She was at McGill, at University College London, at Rockefeller University. I was at McGill, at University College London, at Rockefeller University. And it was a real joy to be able to get to know her when we were neighbors across the street from one another at Rockefeller and Weill Cornell several years ago. So it really is a pleasure and an honor to be here today for the Grafstein Lecture. Today I want to tell you about the wiring and rewiring of the brain, mechanisms through which the brain gets assembled during embryonic development, but also plastic rearrangements that occur in connections later in the adult. The initial wiring of the brain, of course, as you know, starts when each neuron as it's born sends out an axon tipped by a growth cone that navigates through the environment to reach its targets to form synaptic connections. And it's guided to, growth cones are guided to their targets very actively by proteins, guidance cues, located in the extracellular environment. We've known for a long time now that axons are guided through the combined actions of tractive and repulsive cues. And starting in the 1990s, a lot of progress was made in identifying the molecules that function as attractants and repellents, or both. Many molecules are bifunctional. They're attractive to some, repulsive to others. In the 1990s, several labs, including ours, identified the first major families of axon guidance molecules. I like to call them the canonical guidance cues, the members of the Nephron, SLIT, semaphorin, and ephrin families. They account for many guidance events, not all, so the hunt was on for additional molecules. Again, a number of labs, including ours, showed that important collaborators in guidance are molecules we think of more typically as morphogens, members of the hedgehog, Wnt, and BMP family. And again, we and others have shown that other types of molecules, including growth factors and others, can function to guide axons as well. Today I'm going to focus in particular on nephrons and hedgehogs because of recent insights just the past few years in their functions in the developing spinal cord that have deepened our understanding. So in the first part of my talk, I'd like to talk about further insights into the roles of nephrons and sonic hedgehog in spinal cord axon guidance. I'll start by telling you about prior models of axon guidance in the spinal cord, and then focus on recent insights that are obtained from recent tests of the existing model, as well as highlight some open questions on their function. Then in the second part of my talk, I'd like to turn to the adult and talk about structural axonal plasticity that occurs in an activity or experience-dependent manner. Again, I'll give a brief introduction to some prior work, including some work that we did that really motivated our more recent work on the development of a brain clearing method to study structural plasticity. I'll tell you how much progress we made there, and then I'll close the loop by showing how the very same method can be used to help discover effects of defects in axon guidance cues on circuit organization and circuit function in the adult nervous system. So let's start with the problem of axon guidance during development. I'm going to focus on commissural projections where axons cross the midline to connect the left side of the body to the right side. Of course, there are many examples of such commissures in the forebrain here in the visual system. Again, I'm going to focus on the spinal cord, where there are many classes of neurons whose cell bodies are on one side that project axons across to the other side. They're listed here, although there are different functional classes of neurons. The initial trajectory of their axons is actually very similar for all these different classes. And this was first described by the great neuroembryologist Ramon y Cajal, who described so much about the development of the nervous system. He was very fascinated with the growth of these commissural neurons in the spinal cord. Their cell bodies are located in the dorsal spinal cord. They initially grow along the edge, but then they break away from the edge and project directly towards the midline, whereas there's a specialized set of cells called floor plate cells because they're located at the floor of the spinal cord. Cajal was fascinated by this breaking away of the axons, and these observations, as well as others, helped motivate his chemotropic theory of axon guidance. He proposed that axons might be guided by diffusible chemoattractants, and he proposed very specifically that floor plate cells might make a chemoattractant that would help the axons to break away and come towards the midline. Now this shows the spinal cord with modern-day approaches. Here, looking at the growth of commissural axons visualized with an antibody to Robo3, a protein we described a few years ago. You can see this beautiful trajectory of the axons along the edge of the spinal cord, and the breaking away when they get to the level of the developing motor column to make a beeline towards the floor plate. And this is in the mouse, which will be the main organism I'll focus on today, a little bit on the rat as well, at day 11 and a half of gestation. So I want, in the next few slides, just to describe how we initially were able to show that Cajal was right, that the floor plate makes a chemoattractant activity, its molecular characterization, and then build up to some deeper insights that have resulted from recent studies. So historically, we first demonstrated the existence of a chemoattractant for floor plate cells in experiments in which we took pieces of dorsal spinal cord, in this case from embryonic rats, you can do it with mice, and culture them either alone or together with microdissected piece of floor plate. You can see that the floor plate stimulates the outgrowth of commissural axons, showing that the floor plate secretes an outgrowth activity for these axons. It's suggested that the floor plate might be attracting these axons, but if you think about it, this experiment just shows that the floor plate can stimulate outgrowth. We were, however, able to show that the floor plate can attract the axons by taking advantage of the fact that within these tissue explants, if you look inside by labeling the axons actually grow straight, as you can see here in a control situation over 40 hours of culture, so that you can challenge them with a piece of floor plate to the side, and if you do that, you can see that the axons in the vicinity will reorient growth towards the floor plate. So this shows that the floor plate secretes a turning activity for these axons, and we could estimate the distance about 150 to 250 micrometers for the effective range of this factor. So the floor plate can stimulate outgrowth of the axons. It can stimulate turning of the axons. We wanted to identify the factor. We took a biochemical approach to identifying the factor made, the outgrowth promoting factor. We found that extracts of embryonic chick brain possessed a similar activity that could stimulate outgrowth, and using this assay as a bioassay to track the activity, Tito Serafini and Tim Kennedy in the lab were able to purify the factor to homogeneity. We found that the activity in brain extracts was due to not one but two proteins of molecular weight 78 and 75 kilodaltons that were related. We call them Nephron One and Nephron Two, and they show homology to the gamma chain of laminins. Now, in the mouse, which I'm going to focus on, in the rat, only Nephron One is expressed in the spinal cord, and in fact, it's made by floor plate cells. Importantly, it can mimic both activities of floor plate cells. It can stimulate outgrowth, not surprising, that's how we purified it, but importantly, it could also stimulate turning of the axons, as here. So Nephron One is made by floor plate cells, can mimic the outgrowth activity and the turning activity. Does it account for all of the activity? The answer is yes for outgrowth, but no for turning. Let me show you this in the next slide. So here in experiments first done by Tito Serafini, but here repeated by Fred Chiron, you can see that Nephron stimulates outgrowth of commissural axons, or rather the floor plate does, if it's from a wild-type animal. But if you take floor plate from a Nephron knockout animal, you can see that the activity is completely lost. So Nephron accounts for all of the outgrowth activity of floor plate cells. But now if you perform the turning assay, where the axons will turn towards the floor plate, here's the micrograph, here's its trace for clarity, you can see that if you use Nephron knockout tissue, that the axons still turn. There must be a second chemoattractant in addition to Nephron. So Fred wondered what this attractant might be. He used a candidate approach and tested a number of factors, including one sonic hedgehog that's expressed very highly at the floor plate, and in fact turned out to be the second chemoattractant. So sonic hedgehog does not stimulate outgrowth, unlike Nephron, but it can stimulate turning when presented from the side here, just like Nephron does. So floor plate cells, shown here, make both Nephron and sonic hedgehog cause turning. If you take away Nephron, you still have turning. What about if you take away sonic hedgehog? Well, this you can do over here using a pharmacological agent. It turns out you don't abolish turning either. So take away one of the attractants, you don't abolish turning. Take away the other attractant, you don't. What if you take away both by using a Nephron knockout floor plate as well as the pharmacological agent to block sonic hedgehog signaling? Then you abolish attraction. So it shows that the floor plate makes two attractants, which are fully redundant with one another. Block one or the other, you don't compare attraction. Block both, you get rid of this. And so our theory then, elaborating on Cahal's model, is that this breaking away of the axons might involve a chemotropic gradient that involves two factors, Nephron One and sonic hedgehog, which are redundant, at least in vitro, and that in vitro act over 150 to 250 micrometers. With overlapping but distinct activities, sonic hedgehog just causing turning, Nephron causing both turning and outgrowth. Just a little bit more detailing in terms of the receptor mechanisms that mediate the attractive effects of Nephron and sonic hedgehog, and it'd be great if you have another pointer, because this one is becoming very frustrating. And I guess I could use this here as well. But if you have one, that would be wonderful. Now it's working again. Great, thank you very much. Great, thank you. We'll try this for a while. Thanks so much. The receptors that mediate the attractive effect of Nephron, many of you know that, and this was based on insights initially from C. elegans, we found that a protein called DCC is the major receptor that mediates the attractive effect of Nephron One in this case. We realized more recently, a few years ago, using more sensitive reagents, that actually DCC doesn't function alone. It gets an assist from its close cousin, a protein called neogenin, similar structure, high degree of homology. We also found a third protein that potentiates the activity of these two proteins, a protein called ROBO3, which has also an Ig superfamily member, but with a different structure. I don't have time. This is a very fascinating protein. It has at least 3 activities. For the experts in the audience, it can block slit signaling. It can mediate repulsion through a protein we identified called NEL2. But it also functions to potentiate Nephron function. I'll just show you one piece of data here to whet your appetite, and hope you'll go read this paper here, which is, here you see a dose response where you add more and more Nephron, you cause more and more outgrowth from dorsal spinal cord. If you use the dorsal spinal cord from ROBO3 knockout animals, you can see that the response is reduced, and there's a muting of the response at all concentrations, so it's a potentiator. In any case, I'm going to focus primarily on DCC here. In the case of sonic hedgehog, Fred was able to go ahead and identify the receptor complex that mediates the attractive effect of sonic hedgehog. It involves the canonical signaling component smoothened, as well as he showed with Ami Okada in Sumakuno's lab, a binding protein called BOC, also an Ig superfamily member. So these are the players. What then is the role of sonic hedgehog and the role of Nephron in guiding the axons? Well, Fred addressed this in the case of sonic hedgehog by knocking out either this component or that component to look at the effect on the axons. Normally the axons, again, will grow along the edge and then project towards the midline. But if you take away smoothened in commissural neurons, you can see that many of the axons fail to make this turn. They continue to project through the motor column. They will actually eventually correct their trajectory. So you have more axons in the ventral spinal cord here, which is shown in this quantification. The same is true if you get rid of this component here, BOC, where again, instead of the axons growing straight, you can see that many of them will invade the ventral spinal cord again quantified here. So the fact that many still make it to the midline is perhaps not surprising, since there's at least one other guidance cue, Nephron One, that's there. So what's the function of Nephron One? And here things get a little bit more complicated, because it turns out that Nephron One is made by floor plate cells, as I told you. But if you look at nephron mRNA, it's expressed not just by floor plate, but also in the ventral 2/3 of the ventricular zone, here at day 10.5, here at day 11.5 in the mouse. The cells that make this, of course, are progenitor cells, they're radial glial cells that extend the entire length from ventricle to pia. I'm going to use a terminology here that was introduced a year ago by Samantha Butler. So the nephron transcripts in the floor plate are called FP nephron 1, and that in the ventricular zone, VZ nephron 1. This is at the level of RNA. If we look at the level of protein, which was first done by Tim Kennedy in the lab, and then repeated here by Zhu-Hao Wu, you can see at this stage that there is some nephron enrichment in the floor plate, but not dramatically compared to the rest of the spinal cord. A lot of enrichment along the pial edge here, as well as nephron that's visible throughout the ventricular zone here. And again, same thing a day later, highly here, but also along the edge, and also, interestingly, on the surface of the axons projecting towards the floor plate. So what happens if you take nephron away? If you take nephron away, you have dramatic defects. So here's the normal trajectory of the axons. In a nephron knockout, this is a null, you can see that there are defects in the dorsal spinal cord, where some axons, a few axons, wispy axons, will project more medially, but there's also disorganization along the edge and some stalling. You can see profound misguidance in the ventral spinal cord. Instead of making this beeline, you can see that they wander. And so very few actually cross the midline. You have a much reduced commissure. Just briefly, two other defects. There are never any axons growing dorsally here in the wild type, but in the nephron knockout, you see many axons projecting dorsally in the wrong direction, and some that actually exit the spinal cord via the dorsal roots and the ventral roots. So profound effects of nephron, but since nephron comes from two sources, it opens a number of questions. Do VZ nephron 1 and FP nephron 1 both contribute to guidance? Secondly, our theory was that FP nephron 1 made by floor plate cells, would actually diffuse through the environment to track the axons. Does it attract at a distance? And do the two factors from floor plate cells, FP nephron 1 and sonic hedgehog, collaborate to guide the axons? So those are some of the questions that we wanted to address. And indeed, two groups started to address some of these questions directly. The lab of Alain Chidot et al. In Paris, who focused on the hindbrain, and the lab of Samantha Butler at UCLA, who focused on the spinal cord. And what they did was to do region-specific knockout of nephron in different parts of the spinal cord. So remember, nephron is made both by floor plate as well as by the ventricular zone, the ventral 2/3. So using a flox allele, you can delete nephron transcripts just in the ventricular zone using this particular driver that gives what we will call a delta VZ nephron 1 mouse. You can get rid of it in just the dorsal spinal cord using this driver, which gives a delta dorsal ventricular zone nephron 1. Or alternatively, you can leave the ventricular zone intact and get rid of the transcripts in the floor plate using this driver to create a delta FP, delta floor plate nephron 1 mouse, and compare it to the nephron null, which I showed you on the previous slide. So what happens when you perform these manipulations? Well, let's look at the results first from the Chidot et al. Lab in the hindbrain, where the geometry is somewhat different than the spinal cord, and the size also. Here, the neurons actually grow all the way along the edge of the hindbrain, the commissural neurons, to the floor plate. Here you see floor plate transcripts, and in the floor plate and in the ventricular zone, these are nephron transcripts, excuse me. You can see those, that's the RNA, here's the protein. You can see again it's enriched along the edge and at the floor plate. But what happens if you take away either just the floor plate nephron or just the VZ nephron, or both? Well, again, here's the wild type trajectory of the axons visualized with an axonal marker, Robo3. Here, if you take away nephron, profound defects where very few axons cross the midline, as I showed you in the spinal cord. This is now the hindbrain. If you get rid of VZ nephron, you can see that there are also profound defects with very few axons crossing the midline. But in contrast, if you get rid of nephron at the floor plate, there appears to be very little that goes wrong. Here's a quantification just of the size of the commissure here, which normally is dramatically reduced if you take nephron away altogether. This is a hypomorphic allele, so you've gotten rid of most but not all nephrin transcripts. You can see some axons grow. And getting rid of the ventricular zone nephrin gives you a similar effect, not quite as profound as that, but pretty serious. But look here, taking away nephrin from floor plate doesn't affect the size of the commissure at all. It appears, therefore, and they propose that in the hindbrain at least, that the major source of nephrin is the ventricular zone nephrin, and that the floor plate nephrin is actually dispensable, since it had no effect on guidance. Dispensable, by the way, doesn't mean that it isn't doing anything. It could be redundant, for example, with Sonic Hedgehog. Perhaps you'd have to knock out both nephrin and Sonic Hedgehog to see an effect. Also, the VZ here doesn't account for everything, since there's still some guidance that remains when you take it away. But the major picture, though, is that floor plate nephrin 1 is dispensable. That's the hindbrain. What about the spinal cord, which is what we've focused on? Well, here Samantha Butler's lab used this, first looked in the dorsal spinal cord using the Cree driver, where Cree is expressed here. So she will get rid of nephrin transcripts in the top half, but not the bottom half of the spinal cord. Here you see, if you focus on this region here, that in a wild type, the nephrin is shown there in green. The same region in an animal, this delta DVZ animal, you can see the green is all gone in that region. So she's gotten rid of nephrin selectively in the top half of the spinal cord. So what happens if you do that? Well, first, there are defects in the spinal cord where you see some, in this case, in the mutant, some wispy axons dorsally. So the wispy axons I showed you previously may be due to loss of nephrin here. There are some axons that grow dorsally, again, a phenotype you see in the nephrin null. And in fact, there's even a defect down in the ventral spinal cord where the axons are more spread out over the ventral spinal cord. So there's misrouting dorsally, some medial projections in the dorsal spinal cord, and some disorganization ventrally. So this shows an important role for the ventricular zone nephrin, and this is just getting rid of that's present dorsally. What if you get rid of nephrin from the floor plate here, again using this particular driver? Well, she did not characterize this in detail. She noted first that there's no phenotype in the dorsal spinal cord, and that ventrally axons still reach the floor plate, but again, didn't look at it in detail. A major conclusion from this is that there's direct evidence for the importance of nephrin up here in the dorsal spinal cord, although still a question about what it's doing down here. So we decided to investigate this in greater detail, and the we in this case is actually a full collaboration between our lab and Fred's lab. Members of my lab include Zhu-Hao Wu, as well as Sean Teo, Nicolas Rognier, and Olaf Olsen. And in Fred's lab, I think 2 IPN students, Shirin Makihara and Nursen, and I have to get Nursen's name right, Balekoglu. I hope I pronounced it correctly. As well as Patricia Yam, who is a former postdoc in the lab. And we decided to reinvestigate these experiments, getting rid of nephrin in the floor plate, and we used the 2 conditional alleles that were used by the 2 groups, the one used by Samantha Butler, which we'll call F1, and the one used by the Shedotal lab, which I'll call F2. So again, deleting nephrin just in the floor plate, we can ask, and now let's look in more detail in the spinal cord, does anything happen? Again, as was shown previously by Samantha, many axons still reach the midline. So far, so good. But if we look in more detail, we actually see a number of phenotypes. First, a reduction in the ventral commissure, about a 30% reduction in the size of the commissure. Secondly, a change in shape of the trajectory of the axons. Normally, axons come down in a V shape, as you can see here. But in the mutant, the axons come down more vertically, giving more of a U-shape appearance here, which we can quantify with this distance. You see it goes up in the mutant. Actually, Samantha tells us that she sees the same thing in her experiments, where you see actually quite prominently this change from a V to a U shape and a change in the reduction in the ventral commissure. And we saw the same thing in the second allele of nephrin, again, getting rid. Here you can see a very prominent change from a V to a U shape and a reduction in the commissure, again, the quantification in both cases. So there are actually some defects that occur locally, both in terms of the size of the commissure and the final trajectory of the axons, but they're still pretty local. Are there defects at a distance? Remember, our theory was that the floor plate affects the axons at a distance. So for this, we had to look at the axons as they break away and whether they go straight to the floor plate or enter the ventral spinal cord. And so here we're going to look at axons wandering within the motor column, visualized with this marker. You can see that normally in a wild-type animal, there are a few axons in there. But if you take away netrin from floor plate, you can see that we actually increase the axons here. There's essentially about a 50 to 100% increase in the axons that are misrouted and project into the ventral spinal cord. That was true with the F1 allele. The same is true here with the F2 allele, where you also see an increase of axons in the ventral spinal cord. So putting those two slides together, we see three phenotypes. First of all, a reduction in the size of the commissure. Secondly, a change in the shape. We're going from a V to a U shape for the trajectory of the axons. And thirdly, axons aberrantly entering the motor column, providing evidence for action of the floor plate at a distance. Now, that's what happens if you take away netrin from floor plate, but remember our theory was that netrin collaborates with sonic hedgehog to guide axons. So we wanted to ask what happens if you get rid of netrin from floor plate as well as sonic hedgehog signaling. So here, this is what I just showed you, taking away netrin. This is using the F2 allele. This is a repeat of what I showed you previously, Fred's previous experiments, getting rid of the sonic hedgehog receptor Bach, where you can see in each case an increase in axons in the ventral spinal cord, as well as in this case, a change, a more U-shaped, and a reduction in the commissure. What happens then in the double knockout, and what you can see here on the diagram, I hope you can see that there's actually a further increase in the axon misrouting in the ventral spinal cord, as well as additional changes here, and that might be more easily seen in the quantification. Let's start with the commissure, where you'll recall that if you take away netrin from floor plate, there's a reduction in the size of the commissure. If you take away sonic hedgehog signaling, there's no change in the size of the commissure. If you get rid of both, what you have is the effect of taking netrin away. It's an additive effect, if you will. The same thing in terms of the change to the U shape, where we're measuring the distance to the bundle here, which increases when you take away netrin. It doesn't increase when you take away sonic hedgehog. In the double knockout, it's the same as taking away netrin. But now, in terms of this phenotype, the invasion of the motor column, where taking away netrin from the floor plate, there was an increase. Taking away Bach, there was an increase. There, if we take away both, you can see that there is a greater effect of removing the two. Again, the effects seem to be fully additive. And this slide here just summarizes this: reduced commissure, lateral displacement, and increased invasion. We see all three when we take away netrin. We see just one of them when we take away Bach, and if we take away both, we see additive effects. So collaboration between the two, but some differences, and collaboration and differences are reminiscent of what I told you earlier about the in vitro activity, where netrin could cause turning and outgrowth, but sonic hedgehog only turning. Perhaps the difference in the phenotypes relates to these two different activities. That's one of the open questions. Why do we have slightly different effects and slightly different activities? Regardless, this fits with our model that floor plate, Nephron One, and sonic hedgehog are redundant attractants, acting over these distances with overlapping but distinct effects. But you'll note that in this double mutant, there's still some axons that make it correctly to the floor plate. There must be other cues that guide, and a good candidate for this is netrin made in the ventricular zone, which is another source of netrin, and perhaps other cues as well. Fortunately, our friends and collaborators in Paris, Alain Chédid et al., were doing a parallel study where, having studied the hindbrain previously, where they saw no defects, they looked in the spinal cord and did a terrific complementary experiment of, first of all, taking away netrin from floor plate, the same experiment as I showed you in the previous slide, using the F2 allele, taking it away from the ventricular zone, which they did using this particular construct, and then putting the two together, a double knockout, if we will. It takes it away from the floor plate and the ventricular zone. This is a good control because it should phenocopy the full null. It should be the same as taking away netrin away by itself. So what happens in these three situations? Well, again, the axons normally grow to the midline. Very few of them enter the motor column. None of them grow dorsally. If you take away netrin from the floor plate, you see the same phenotypes as we observed in the previous slide. You see this U shape here, very prominent. A reduction in the size of the commissure, that's shown here, a reduction in the size of the commissure. And also invasion, you can't see this very well with the micrograph, but you can see it over here with the quantification that more axons invade the ventral spinal cord. So again, same results as we had in our previous slides. But in this case, what happens when you take away netrin from the entire ventricular zone but leaving it at the floor plate? You see a number of things. First of all, you see increased invasion of the ventral spinal cord. So
VZ netrin does contribute to guidance down here as well. You see this dorsal migration of axons, so that's attributable to the ventricular zone. You also, but you don't see a change in the size of the commissure. So you see this increased invasion of the ventral spinal cord. Important control, if you do the double knockout of taking it away from the floor plate and the ventricular zone, you predict that it should be the same as taking it away altogether. And that's, in fact, exactly what happens, showing that these constructs really do the job perfectly. You can see, again, a complete reduction of the commissure here, and also further invasion of the ventral spinal cord. Note that the scale here is greater than the scale here. This, again, is a supra-additive effect. The two, taking away the two, provides more than an additive effect of taking away either alone. They function redundantly. So from this, we can conclude that unlike in the hindbrain, in the spinal cord, netrin from floor plate and netrin from the ventricular zone are both important. Midline crossing, floor plate netrin 1 is essential. In the dorsal spinal cord, to avoid that dorsal migration, the ventricular zone netrin is essential. And in the ventral spinal cord, there's partially redundant contributions of FP and VZ netrin 1. So again, our model then was that these two function to attract, but there are new players now. Ventricular zone netrin in both the dorsal and ventral spinal cord also contribute to the active guidance of these axons. Well, there are a number of important issues that come up from this. First, what's the difference between the hindbrain, where there are no defects, and the spinal cord, where there are defects when you take away netrin from floor plate? One difference between the hindbrain and the spinal cord is geometry, where in the hindbrain, axons simply grow straight. They never have to break away from the edge. It's not clear what you would use a chemoattractant for if you were designing the system. So perhaps that helps explain why it's less important. Another difference is the size or distance. Remember, we're postulating that these chemoattractants act over 150 to 250 micrometers, this distance here. If we show the hindbrain at the same scale now, where we match the scale bars, here's the hindbrain early, here's the hindbrain late. You can see that the distances are much vaster. So again, the system seems to be set up primarily for guidance along the edge to the midline. Again, less clear why you'd need a chemoattractant. So it supports a model in which these attractants act over a distance of 150 to 250 micrometers, and they're primarily used for reorientation when the geometry is appropriate to the task, not necessarily used in every case of axons projecting towards their target. A second issue, we've postulated that netrin from floor plate diffuses at a distance. Can we actually visualize that diffusion? And what about netrin from the ventricular zone? Well, here Tim Kennedy, years ago, obtained some evidence on this point because of a quirk of nature, which is that whereas in the mouse, netrin 1 is made by both floor plate and the ventricular zone, in the chick, there are two netrin genes, netrin 1 and netrin 2, and they each perform half of the role. So netrin 1 is just in the floor plate, and netrin 2 is just in the ventricular zone. So it's like you've taken apart FP netrin and VZ netrin just through a natural experiment. So Tim made antibodies to netrin 1 and netrin 2 that were selective, and what he found was quite interesting, which is that when you look at netrin 1 protein, you can find it at a distance from the source, about 150 to 200 micrometers. It fits. Netrin 2, interestingly, seems to be stuck at the level of where the transcripts are. It's as if it's not as diffusible, and supporting the idea that netrin coming from ventricular zone cells, this could be a difference between netrin 1 and netrin 2, or it could be a difference between floor plate and ventricular zone. In the mouse, where again, netrin 1 is both in the floor plate and the ventricular zone, if you look at netrin 1 protein dorsally, where it's made from the ventricular zone, you see that it does not diffuse away from the sites of transcript expression. So the evidence suggests that netrin made from floor plate is diffusible, but from ventricular zone is not. And further evidence for this came from early experiments where we microdissected lots of different pieces of embryos. We found that floor plate always has outgrowth and turning activity very strongly, but if you take pieces of dorsal spinal cord, they don't have activity in these assays, which require action at a distance. And from ventricular zone, where there's lots of transcript, there's very little, if any, activity as well. Again, supporting this idea that this netrin here, made in the floor plate, is diffusible, but here is not. So how does this arise? Well, maybe the protein is modified in some way when it's made here versus in the ventricular zone. That helps explain the difference in diffusion. Perhaps there's a specialized secretion mechanism. This is something that Samantha Butler and her colleagues proposed, that perhaps the radial glia in the ventricular zone transport the netrin down. Remember, it's enriched around the edge. You'll recall the transcripts are here, the protein is enriched there. Perhaps it's transported down and deposited there in a way that makes it less diffusible, clearly something that needs to be explored further. Last issue I want to raise here, which is how is it that netrin is guiding? In the dorsal spinal cord, there's a corridor of netrin, and the axons initiate growth down the corridor. Here you can see the axons within the corridor. Perhaps the corridor helps funnel them. If you look more carefully, as Tim Kennedy did, there's actually, the netrin is present in a gradient. He could quantify a dorsal to ventral increasing gradient of netrin protein along the edge, and then through the ventral spinal cord here, experiments done in the chick embryo. Just because there's a gradient there doesn't mean that it matters, so this is something that could be tested by modifying the gradient to ask whether that's what's responsible for guidance. But in any case, the data that I've showed you are consistent with the idea that local guidance by Nephron One, made by ventricular zone cells, involves enrichment of netrin along the edge that provides guidance locally here and probably in a non-diffusible form for the netrin. But the data do support the original model that Nephron One made by floor plate as well as sonic hedgehog can travel away and attract the axons at a distance, consistent with Cahal's idea. Many open questions. Is it true that floor plate Nephron One is diffusible and VZ Nephron One not? How is it that guidance occurs? Is there a corridor mechanism? Do gradients, are they important in the dorsal or ventral spinal cord? Issues related to mechanisms, and of course, they are not the only cues. There are others as well, but in the interest of time, I'm not going to talk about that. And I want to, before I close this section, I do need to mention, of course, that although we focused on this in the system of the spinal cord, we believe that this has relevance much more broadly, not just to rodents, but also to humans, starting with the beautiful work of Myriam Srour and Guy Rouleau and their colleagues. It's been known for some time now that there are mutations in humans in Nephron One, in its receptor DCC, and also in this potentiator, ROBO3, that give rise to a number of inherited neurological disorders. In the case of DCC, this original study was congenital mirror movements, which I'm sure everybody here is familiar with since the work was done here in Montreal. So in the last part of the talk, I want to turn now to structural axonal plasticity in the adult brain, to go from development to the adult, although I'll come back at the end. And I'm really going to describe briefly some work in progress. First, a little bit of motivation for why we've been interested in this. We've been focused on axon growth and guidance and branching in the embryo, but it turns out that axons can also be reawakened and caused to grow in the adult. This was first shown by Mike Merznick and his colleagues at UCSF about 30 years ago, working in primates, where they showed that sensory deprivations in the periphery could trigger compensatory changes in the cortex, so that cortex that essentially is silenced because of loss of sensory input gets innervated from neighboring regions. It's sort of a filling in effect of axons. That was shown first in the primate. It's also been shown in rodents, and the system that we've been studying is in the barrel cortex of mice. This is a system that's been set up by Charles Gilbert and his colleagues in this type of paradigm. And we collaborated with Charles when we were at Rockefeller University. Charles is on the faculty there. I just want to remind you that the major whiskers of the snout of rodents has a representation in the cortex called the barrels. Each of these whiskers is represented by an individual barrel. So if you move the whisker, you'll see electrical activity in that barrel, but not the others. It's a system that shows a functional plasticity that's well known. If you shave or pluck whiskers, for example, the D rows and the E rows, so that the D and E barrels are initially silent, what you see is that initially they go silent, but then as you wait over time, within a day and several days, you'll find that these areas of the barrel cortex get activated when you move the C whiskers. There is functional compensation. And what Charles and his colleagues have shown is that this may involve just modifications of pre-existing circuits, but there's also evidence for axonal sprouting and remodeling that occurs in response to this sensory silencing. What I like about this system is the injury or the manipulation, if you will, is done here. There's no actual injury or inflammation here. This is really an effect that occurs as a result of silencing, and what you see is that some neurons will sprout axons to innervate the neighboring barrels, essentially to activate them. Nature abhors a vacuum. The reciprocal experiment can be done, and here I'm just showing work from Randy Bruno's lab, where they showed that if you look at axons within a barrel and you silence that barrel, that there's actually shrinkage of the axon terminals. Here you see some quantification. So you can get axons to sprout into silent areas, or axons in silent areas will shrink down. We became interested in this through our studies with the Gilbert lab in a type of paradigm where they create a cranial window over the barrel cortex and inject virus into neurons in a barrel, then deprive the neighboring barrel. Normally, the axons that project from one to the other are completely static, but if you deprive a barrel by plucking or shaving a whisker, what you'll see is very rapidly axonal remodeling that involves both sprouting, shown here in yellow, as well as retraction. There's a remodeling of the axon that continues over a matter of days. They had established this paradigm, and we were interested because we were studying a mechanism of axon pruning involving a receptor called DR6. And I'm not going to go through the details of this, just to say that in the DR6 knockout animals, we knew that in development there were defects in pruning, and we were gratified to see that in this model here of sprouting and pruning induced by sensory deprivation, that there was a reduction in the pruning. There was less red here in the pruned axons. And here's just a quantification that normally there's quite a bit of pruning that's shown here with the dotted line in the wild type after sensory deprivation. But in the DR6 knockout, you can see that this is much reduced. And again, without going through the details, because that's not the purpose of this segment, we have been focusing on a pathway that involves interaction of DR6 with the amyloid precursor protein and a downstream pathway of degeneration. And again, in the APP knockout, we could also see a reduction in pruning, less red here after sensory deprivation in the knockout compared to the wild type. So some evidence that APP and DR6 are both involved, and in fact preliminary evidence that downstream components are as well. Again, in the interest of time, I don't want to go through the details of this pathway. This was interesting, but it got us very frustrated because these experiments are incredibly difficult to do. It takes a long time to follow a single animal and a single cell or group of cells in an animal. And we wanted a more facile way of being able to study axonal rearrangements that would permit more robust analysis and especially genetic analysis. So that was really the motivation for us to do the studies that I'm going to describe now. Which was to try to leverage brain clearing methodologies that had started to come online about the time that we were doing these studies. I think you're familiar with these, and in the between about 2011 and 2013, 6 different brain clearing technologies were developed. One that's well known by my colleague Carl Dijsselhoff at Stanford called Clarity, but there are others as well. And we were excited to use those methods in order to study axonal rearrangements in the adult nervous system. And if you'll bear with me, I hope the logic of this will become clear in a few minutes. This just shows you the challenge. Here is the largest embryo on which you can do whole mount immunolabeling using standard techniques. You can see the brain, these are to scale. The adult mouse brain, not even a human brain, but the adult mouse brain, you can see is much larger and of course much fattier because of all the myelin. So you can't, with standard methods, do immunolabeling. It turns out that what these brain clearing methods have in common is that they delipidate the brain and then they match refractive index. So you get rid of the lipids and you then can make the remaining tissue transparent. And we were excited to see whether this could be used for what we were interested in. It turns out that the existing ones were not that good for maintaining epitopes, biochemical epitopes that we wanted to label by immunolabeling, for example, cleaved caspase or others. Most of the delipidation methods are very harsh and destroy epitopes. They will, in some cases, preserve GFP fluorescence, but
are not as robust for immunolabeling. But we were able to take those methods and develop a new method that combined ideas from some and added a little bit more that we called iDisco. I'll explain why in a minute. It starts by taking this adult brain and then performing a gentle delipidation that makes the brain porous enough that we can get antibodies to go in for immunolabeling. We then, once that's done, we can do a complete delipidation and then clearing of the technique and refractive index matching. And if we do this, we've preserved epitopes for this gentle delipidation. We can get very good immunolabeling. So this method is good. It works in the adult mouse brain. It preserves morphology, importantly. Other brain clearing methods have difficulty there. I'll explain why that's important in a second. It's compatible with a wide range of antigens, which is what we wanted. So it checked those 3 boxes. It doesn't preserve GFP fluorescence. That's one downside of this method I'll come back to later. That's because this clearing method, which is very effective here, actually kills GFP fluorescence. But we can come in with antibodies to GFP if that's what we want. This just shows you the obligate clearing slide. Here's an uncleared brain. Here is a cleared brain. So let's use this in the barrel cortex, and what we started with, what we were doing really as positive control experiments, but that turned into an interesting series of functional plasticity experiments, where we wanted to look at whether we could use the barrel cortex to study axonal rearrangements, but we first asked whether we could use it to study other changes. And we decided to look at immediate early genes like CFOS, which, as you know, are surrogates for electrical activity. So imagine in a situation where we've shaved all these whiskers so that only the B row whiskers are still present. If we then activate them, you expect there to be more electrical activity here. You predict that CFOS should be expressed there. We should be able to detect that with an antibody. And sure enough, the iDisco method is compatible with that. So you can see that CFOS here is expressed in just the B row in this experiment. And the same is true of these other immediate early genes like ARC, EGR1, and so forth. Here they have different patterns, interestingly, something that's harder to see if you cut sections. In the background here, you see tissue autofluorescence. That's in a different channel, which shows very beautifully where the barrels are located. Let me just show you what this looks like. This is a movie. We take the whole brain and we use a light sheet microscope to get optical sections through the tissue. We also developed software so that we can the computer can identify the FOS-positive cells. Red is the immunoreactivity, so that's the actual data. At the center of each of those red spheres, there's a little green dot which has been detected by the computer. The computer's detected the cell. Here you just see the computer's extracted all of the cells, and we can then go to a different channel and see this tissue autofluorescence from some residual lipids in the brain, which we use to register the brain to an atlas, the Allen Brain Atlas. Here you see the barrels in autofluorescence. Now the green is turned to white. These are the cells expressing CFOS. We'll get rid of the autofluorescence. It's pretty dark, but as we reduce, hopefully, the magnification, you'll see that here, these are C-row spared. The C-row is very clearly evident. We've actually developed software to take the light sheet data sets and map them to the Allen Brain Atlas, so they're registered to the atlas. We can detect the cells, we can count them, and because it's registered in atlas, we can now average across regions. We can actually get averages and standard deviations to actually do some statistics, all of us in an automated fashion. So for example, here are 3 animals where without any manual adjustment or anything, we did iDisco to identify the FOS-expressing cells and registered the Allen Brain Atlas, got the average, used a heat map display. Here you see where the cells are active. Here are 3 animals where it was the A row that was spared, here where the B row was spared, where the D or the E row. You can see that it's a very powerful method, again, with automatic isolation of the relevant part of the cortex. This enables us to do experiments like this one, where we shave whiskers, and we have chronic deprivation experiments looking at what happens to activity in the the barrels that still receive input and the neighboring ones that have been silenced at 1 day and up to many weeks. So at 1 day, again, here's no trimming. There's activity in all the barrels. After 1 day, we see, in this case, it's a C row spared activity in the C row, but you can see that after a week, there's compensation, where now we see spreading of the activity to other barrels, and actually a change in the patterns of activity that are evident as well. This is just projecting that part of the cortex, but of course we have the full data sets. We can take the same data sets and actually look with other types of views. Here, again, in this sagittal view, you can see that the C row is highly active at 1 day after trimming, with the neighboring rows silent. But you can see 5 weeks later, this is extended. And if we want to look for new areas of activity, the most powerful, since we can do statistics, is to actually look for areas where changes in activity are statistically significant. We call this a p-values map, where in green we show any regions where activity is statistically significantly different at 5 weeks versus 1 week, where it's higher, or where it's lower, we show it in red. So here you see areas that are of higher activity at 5 weeks. Here's another region where we see more activity in the supplemental somatosensory cortex. If we look in the thalamic nuclei, by contrast, you can see it's all red because the activity is lower in a statistically significant way. So we have this powerful method then for visualizing data sets and mapping them to the Allen Brain Atlas. Let's get back to axons, and this is now leading up to the last part. The problem is that we would like to look at axons projecting, for example, at just one barrel, and then ask if we deprive the neighboring whiskers of whiskers, and then we silence these barrels, is there sprouting of the axons into the neighboring barrels? The problem is that there is no promoter that will enable you to label just those axons. So what we've done is to trace axons in an activity-defined subcircuit using a mouse generated by our neighbor Li Qingluo, where a Cree ER is actually made under the FOS promoter, actually as a fusion. So if a cell expresses FOS, it's electrically active, Cree will be present. If we provide a reporter to the same cell and tamoxifen, we can indelibly label the cell that was active at that time. So we can shave all the whiskers except for one. We will get FOS activation along the circuit from the whisker through the thalamus all the way to the cortex. If we play our cards right, we should be able to label just those axons and then allow the whiskers to regrow. We inject the reporter down here in the thalamus. And then we can retrim. And in this case, if we've labeled the axons projecting to that barrel and trim again so that that's the only active barrel, you might predict that the axons will sprout outwards to the silent barrels. Vice versa, if you label just that barrel but do the opposite of shaving only that whisker, so it's silent, then as in the Randy Bruno experiment, you might expect the axons to shrink. Or my favorite, if you just label one row, here the C row, and then you come back and you silence just two of the rows up here, you might predict that the axons would sprout more towards the silent than towards the non-silent rows. So you can see that this paradigm should be very powerful if we can get it to work. And can we get it to work? The answer is yes. You can see that we can label these cells projecting just, in this case, to the C2 barrel using this trap and trim paradigm, or to all of the cells projecting to the entire C row if we spare the entire C row. This is where we've gotten recently, and we are starting to have some initial evidence for structural plasticity. In this type of paradigm, you predict that the axons might sprout to the neighboring barrels. Here's an untrimmed animal, and here's now a trimmed animal. You can see that there is indeed an increase in the sprouting of the axons to the side. So this is now our program of analysis, which is to trap axons projecting to a single barrel or a row and perform these types of manipulation to understand the logic of this. But the beauty of it also is that when we deliver the reporter to the thalamus, we can put in, for example, guide RNAs to knock down particular candidates, so we can actually use this in a more facile way. So the technology is now in place for us to actually begin to understand the logic of sprouting in the adult and also its molecular mechanism. Just a few more points. First, we can, in an automated way, identify cells expressing CFOS. We have been extending this to automatic extraction of axons and quantification. I won't go through the details. Together with collaborators at Stanford, we've also been focusing on automated comparison between 3D pathology and optogenetic fMRI together with this iDisco method. We've focused on trying to modify this method so that we can overcome this limitation of killing GFP without going through the details. Zhu-Hao Wu in the lab has now developed a method that is as powerful as iDisco on these 3 parameters, but also preserves GFP fluorescence. And here's an example of that GFP fluorescence preserved in this way. And last but not least, I wanted to bring it back to axon guidance and show how this method can be used to study defects in brain circuitry and function that result from defects in axon guidance molecules. And as a test bed for this, at the urging of Guy Rouleau, we decided to focus on DCC haploinsufficiency, which is known through the work of the Kennedy lab, as well as in mice, as well as a number of labs here in Montreal in humans, to alter the organization of dopaminergic circuitry in both mice and humans. There is a blunted response to amphetamines in mice. There is a decrease in novelty seeking in humans, and I encourage you to read these beautiful papers, which you probably already have since they were generated here. Guy suggested that we try to use iDisco to look at this, again, using the mouse model. So here's a very simple experiment, a small cohort of mice, wild type and DCC heterozygous animals, and some treated with saline, some treated with amphetamine. And the protocol is to inject it, an hour later sacrifice, and do iDisco tissue processing, where again, we're going to look initially at CFOS labeling to look at electrical activity. So let's look at wild type animals first and treated with amphetamine. What I'm going to plot here are volcano plots. This is the full change in CFOS positive cell counts per brain region, so with amphetamine versus without. So here, twice as many, 4 times as many, and so forth, with amphetamine versus without. And what's shown on the vertical axis is the p-value for the change. So there are changes that occur. There's a lot of scatter. Each of these dots represents a different brain region identified by the Allen Brain Atlas. But you can see we identify a set of regions where CFOS increases in a statistically significant manner, all automated. Here we're looking at this comparison in the DCC knockout, saline versus amphetamine. You can see the main picture that emerges is that there's a blunting of responses pretty much in all regions. You can see here the non-statistically significant, but also the statistically significant. And now let's look at this comparison, which is the wild type versus the DCC het, where again what we see is a decrease in activity in the DCC het as is seen in humans. And each of these dots is a brain region that's shown here, so we can identify them either by looking in tabular form or by mapping them to the brain, where again we use this p-values map, where these decreases here in expression are shown, but statistically significant, are shown as red regions here, so it facilitates the visualization. This is to look at activity. We can also look at axonal projections, where again it's been shown here that in the DCC heterozygous mice, there are increased levels of TH protein and also of innervation of the medial prefrontal cortex. We can see this again in our data sets, now labeling the axons for tyrosine hydroxylase rather than looking at CFOS expression. And our automated axon extraction pipeline, we hope, will facilitate the analysis of defects in projections in these mice as well. So in closing then, I hope I've convinced you that this method holds potential for studying axonal rearrangements in the adult, which is what we set out to do. A collateral benefit is that we've been able to use immediate early gene expression, CFOS, to look at activity. And we're excited now to look in axon guidance mutants, and especially in cases like these haploinsufficient mice that have no obvious behavioral deficit just by themselves, to look for more subtle changes in behavior and connectivity, but in a more straightforward way, thanks to all the automation and the facility of looking at the entire brain. So I'd like to close by thanking my collaborators. Many current and former lab members participated in the studies on spinal cord axon guidance. I want to highlight today the work in our lab from Zhu-Hao Wu, Nicolas Renier, Shantio, Olaf Olsen, and also the collaboration, of course, with Frédéric Charon-Chirin, Patricia and Nursen. The work on adult plasticity, again, the people who are involved, and a lot of the work, recent work I described is the work of Eliza Adams, a graduate student in the lab, together with a technician, Anna Maria Sola. And here, just leave you with the current lab at Stanford. Since I'm often asked this, the lab is open for business, and I am recruiting postdocs, so if there are students here who are interested, please don't hesitate to apply. So with that, I'd like to close. Thank you for your attention. I'd be happy to take questions. Thank you very much.
Thank you, Mark, for this fantastic talk. So, time for questions now.
Just one in the back there. Yeah.
I don't know if I don't have a microphone, so I'll speak loudly. So I always thought I understood a little bit of plasticity was done, but I was kind of intrigued by the experiment done by Marc Tessier. I don't know if you know the work he's done on amblyopia. So you have a lazy eye and a good eye, and he injects both eyes with tetrodotoxin, and then boom, 2 days later, both eyes look normal. So how can that be? I mean, there's no activity involved, at least.
I could speculate, except I don't know the experiment and the details of that particular experiment. So it's certainly sounds wonderful and miraculous to me, and I'd love to know more. Yeah. Yes.
Thanks for a great talk. Is it safe to say that the diffusible netrons are phylogenetically younger than the non-diffusible ones?
That's so that you would start with netron that doesn't diffuse, and that there's then netron that diffuses. It's interesting. If you look in Drosophila, so netrons are found in C. elegans and Drosophila, and nematodes and Drosophila in pretty much every animal that's been looked at. Every animal that's been looked at. In organisms that are smaller, you may not need netron to diffuse. There are some experiments in C. elegans that suggest that netron may be acting over a distance of perhaps 40 micrometers. These are some experiments in Corey Bargman's lab by Carrie Adler, who was doing visualization of axons that normally project towards a netron source, UNC6, and in an UNC6 mutant. And what she saw is that as soon as the axon starts growing, the phylopodia are projecting in the right direction. In an UNC6 mutant, she finds that from the get-go, even though it's far away, far in a nematode sense of about 40 microns, it doesn't project. So that's suggestive of a possible effect at the distance, although there are perhaps other explanations. In Drosophila, there's a very nice, you can see a little bit of both. So Barry Dixon did a very nice experiment. He created a construct of netron that, rather than being secreted, so netrons are secreted, but they're highly basic, so they interact with the extracellular environment, particularly with proteoglycans. When we purify them, we found if you express netron in cells, I showed you them having an effect at a distance, but if you actually ask where's the netron, the majority is stuck on the cell surface and only a minority diffuses away, and it sticks to proteoglycans and other negatively charged moieties on cell surfaces. So how far you will diffuse will depend on how many proteoglycans there are in the environment. That's number one. Barry Dixon created a netron that is not secreted, but has a transmembrane domain, but that's still functional. And when he replaced the netron gene in Drosophila with that one, he found that axon growth to the midline was actually normal. So in Drosophila, where the distances are even tinier, perhaps 20 microns, the netron doesn't need to diffuse. The axons can probably reach out and touch and just stick there. Interestingly, in the very same experiments, he showed their netron is bifunctional. There's some axons that are repelled by netron, and those ones were and their cell bodies are like this, they were no longer repelled by the netron that was transmembrane. So it showed that for attraction in that system, local contact was sufficient, but repulsion was required, a diffusible form of netron, excuse me. So If you take those two examples, there's perhaps some tentative evidence for action at a distance, a few tens of micrometers, if you will, in those species. If you go back even earlier, where you find that Nephron first was immobile and then later acquired more mobility, it's possible. It's certainly not a crazy idea. In the spinal cord, there really seems to be a lot of Nephron that's churned out, and it does seem to be quite diffusible. That's why we're quite interested to see whether there are differences in the proteins that are made or in the secretion mechanisms. Yes?
So where do you guys stop? So what are your thoughts about the role of these guidance cues in the adult in terms of this diffusible versus, do you think everything is short range?
Right. So again, what I think when we set out on years ago to study guidance in the spinal cord and found this diffusible activity, we found that the range of action was always a few hundred micrometers at most. And actually, that's perhaps not a coincidence, because if you solve the diffusion equation, under the best of circumstances, it's hard to establish any kind of gradient over more than a few hundred micrometers, a gradient that can be sensed by a cell. I think the use of the term long range, it's longer range than contact, but I think some people may have taken it to mean millimeters, centimeters, kilometers, and so I think it's very important. One of the things that all of this analysis reaffirms is that what we're talking about is distances of about 200 to 250 micrometers. In cases where axons are growing much longer distances, they are not being attracted by something very, very far away. They're more likely to grow along an axon tract or down a corridor. When they get close, and again, our idea is that where the geometry warrants, for example, in the spinal cord, you have to break away from the edge, that there a diffusible chemoattractant could be particularly valuable.
Thank you for a wonderful talk. So when you do the systematic knockdown of nephrons into straight versus maybe like a zone, what happens to gradient of other repulsive and attractive cues? And how is nephrin alone contribute to the phenotype or
Yep.
other factors contribute?
Yes, so that's a great question. So we did look, we looked at cell type specific markers to make sure that, so first of all I should say, the nephron knockout and the nephron null have been examined in some detail by a number of labs to ask whether there are defects in the expression of other cues, and you can never be completely exhaustive, but what's been looked at so far, there are no changes. So there's no evidence that when you take away nephron from the entire spinal cord, that you reprogram the cells or things like that. When you take away just from floor plates, in these experiments, and this was done actually by the colleagues in Fred's lab, there's no change in a series of cell type specific markers in the spinal cord. Now we haven't looked at the gene expression of other axon guidance cues. In principle, they could be affected. At this point in time, there's no evidence that when you alter nephrin, you affect the expression of other guidance cues in the spinal cord when you take it entirely away. So it's not clear why it would happen if you take it away just from the floor plate, although in principle it's possible, so it certainly would be worth looking at. I think it's a strong hypothesis that the effects that we're seeing are really due to loss of nephrin itself, rather than some secondary cascade that results from that.
Thank you for a great talk. So I was wondering about motor neurons. You mentioned
Yeah.
a few times, and sort of appear to be innocent bystanders, but are they really, could they be contributing something to visceral axon guidance?
Right. So the commissural axons break away and skirt the motor columns. The motor neurons are mostly avoided by the commissural axons. You saw that one of the defects that we see is an increased invasion of the motor column. So could the motor neurons actually be actively repelling commissural axons, for example, pushing them away? We certainly believe that might be the case. A few years ago, Alex Jaworski, when he was a postdoc in the lab, studied this protein Robo3, which I used as a marker in my talk. But it turns out that Alex looked for ligands for Robo3 because
No ligands for ROBO3 were known. And biochemically, he was able to isolate, using the ectodomain of ROBO3 as bait, he was able to isolate a ligand, a protein called NEL2, which is a secreted factor. It has very traditional domains of secreted factors, but its function wasn't really known. It turns out that NEL2 will repel commissural axons via ROBO3. So if you present NEL2 to commissural axons, the axons are repelled away. If you take away ROBO3, they don't sense it, so it seems to be a repellent. Where is it expressed? It's expressed by motor neurons. So there's a repellent for commissural axons that is expressed by motor neurons at least at the transcript level. So you say, oh, well, great. What happens if you knock out NEL2, which Alex did? And what we found, we were at first depressed, but then we got cheered up a bit. The depression was that in just the straight knockout of NEL2, there was no defect in commissural axon guidance. Now remember, there are a lot of other things that are guiding there. There's sonic hedgehog, there's floor plate Nephron One, there's ventricular zone Nephron, all of which are contributing to the guidance. He did find that if he crossed the NEL2 knockout to a ROBO3 heterozygous, so just weakening that a little bit, that he then had defects of the axons projecting through the motor column. So we could see in a sensitized background, he could see a defect. But the question is whether, I'd rather see if we take away sonic hedgehog in NEL2, do you see something? If we take away floor plate Nephron in NEL2, do you see something? So we have a candidate for a cue that might be keeping them out of the motor column. It's clearly a very overspecified system, right, where there are multiple cues, all of which are working together to make sure that the axons don't go to the side. Remember, if they go to the side, axons can actually leave the spinal cord. I showed that in the Nephron knockout. So it's probably important to make sure that they keep going towards the midline. But that could be a contribution of motor neurons.
One more question. Do you have an idea of what accounts for the V versus U shape?
That's a great question. So normally the axons come down like this, and when you take away floor plate Nephron, now they go like that. And interestingly, in the Nephron knockout, you saw that the axons are everywhere, but there's no hint that you're sort of more U than V when you take away all Nephron. And so it's reasonable to suppose that you get a U when you still have VZ Nephron, but no FP Nephron. One way to think about it could be that VZ Nephron is distributed in a way that gives you that kind of U shape, and that the floor plate Nephron actually then pulls things in further. That could be a way in which they collaborate. Now, we'd love to know if that's the case, and this is just a little bit more detail. If you look in detail at where the transcripts are, very high levels in the floor plate. I showed you there's the Nephron in the ventricular zone. There's actually a quite high level in the ventricular zone just dorsal to the floor plate. Right? So these are ventricular zone cells. They'll span the distance to the pia. They have higher levels of Nephron than their brethren further up. And maybe that is helping bring the axons, but just not quite enough. And the floor plate Nephron then does that. So that the collaboration between the two is important. Importantly, by the way, if you get rid of VZ Nephron, floor plate Nephron by itself does a lot of good guidance. So that's really the strong evidence that floor plate Nephron is acting at a distance as well. But it's something about this interplay of VZ and FP Nephron that determines whether you're more V-shaped or U-shaped. And we don't know the details. I think looking more carefully at Nephron distribution in the floor plate the delta FP Nephron, or the delta VZ Nephron, will be important to address that question.
Just to comment on that, I noticed in the full knockout, the cells just dorsal to the floor plate are distributed more widely, less concentrated towards the ventricular zone. I think NKX 2.2 cells specifically. So I think maybe the distribution of these cells might be some way influencing the morphology of the commissures.
Again, the colleagues in Fred's lab have looked at a number of markers down near the floor plate, and perhaps there are subtle changes that could explain that. Sort of at a gross level, the general pattern, number and pattern is normal, but you're right, some modest differences could make a big difference in terms of that pattern.
So on this, join me in thanking Marc.
If I could just bear on your and impose on a little bit more of your patience while there are a number of people that I'd like to thank for bringing this all together. So effectively from within our IPN team, I'd like to thank Ed, Ruth Azer, and Fred for actually scaffolding the work, getting Marc on board to come in today. And of course, most importantly, I'd like to thank Debisha Kolehanathan for really doing all of the Joe jobs, which probably should have fallen into my lap for bringing this together. But Debisha's taken care of that and done a wonderful job, and I'd really like to thank her very much for making this event the success that it's been. And also one last thing is I see Sal Carbonetto here in the audience, and I never did thank Sal for actually being the person who introduced Bernice to the IPN. So thanks for that, Sal. It's very much appreciated. We'd also like to thank the MNI Development Office and the McGill Advancement Office, in particular Amanda Fritz, Alexis Gateman, and Anne Charbonneau for all of their fine work in terms of allowing this lecture to come into fruition and their continued support. And of course, last but not least, I'd like to thank both Bernice and Marc for their support, motivation, and inspiration for the current generation of neuroscientists here at McGill and in the Montreal area, and of course, more importantly, the future generation of neuroscientists. Clearly, you have both been phenomenal role models here, and we appreciate that. And as a memento of that, particularly from our students, I'd like to invite Sienna Drake back up to the audience to have a little memento of our appreciation. So if I could invite Bernice and Marc back up to the front stage, please. Last but not least, there will be refreshments served out in the atrium, and we are very happy to invite everyone to come in and participate and enjoy. Thank you very much again for coming.